Vivekananda Centenary College, Rohora, respected Professor Joe Otsuki, Professor Bimal Krishna Bonik, Dr. Bishwadip Banerjee, Dr. Niladri Patro, learned faculty members from different academic and research institutes, research scholars and student participants, and my dear colleagues. Today, we have assembled here to participate in the one-day international seminar on emerging horizons on chemistry. This seminar is being organized by Department of Chemistry of this college. On behalf of the organizing committee, I extend a warm welcome to you all for coming here. Chemistry is often called the central science which connects physical science with life science and engineering. Today, we have gathered not just to discuss molecules and reactions, but we hope to listen from our distinguished speakers and delegates about the inventions which have the immense potential to shape future of our planet. Today, we have amongst us some very distinguished researchers working actively on diverse fields, and I hope that this platform will spark new ideas, new collaborations, and will inspire our young researchers. In this inaugural session, we will begin it with lamp illumination. And for this, I request our revered Principal Maharaj to kindly come forward. And I also request the convener of this seminar, Dr. Taposh Ghosh, who is also the head of the Department of Chemistry of this college, to assist our Principal Maharaj. I now request our Principal Maharaj, revered Shami Kamalasthanandaji, to deliver the welcome address.
So, the most respected delegates in this present convention, especially Professor Otsuki, Professor Bonik, and Professor Patro and Professor Banerjee, and other distinguished faculty members, those have joined from different institutes, and my very dear students in this college and also from outside institution. So this today's one-day seminar, it is meant for the emerging trends of chemistry. So all the chemistry people, all we have know, if we look the human development and the civilization, chemistry plays an important role in every steps. But previously, suppose in more than 20 years ago or 10 years ago, chemistry, what we have learned, that is more or less trial and error methods. But nowadays, the paradigms has been shifted regarding the study and also the investigation in the field of chemistry. Especially the three aspects, that is the integrated digital technology, another absolute this molecular sustainability, and the precision atomic engineering. These have changed the whole study field of the chemistry. Now the present and the future day chemist, they have immense opportunity in the field of chemistry. Our learned speaker, they will enumerate all this, but I can just in my mind what is coming, especially the four domains, this digital and AI technology intervention, green and sustainable mandate, third especially the nanotechnology and precision science, and fourth the future day biomedical device. So these are the main groups, and also others also, and it comprises many subgroups. So these chemistry people, so you all have to understand present day and future day. So you have to equip one or more subdomains for venturing in this emerging field of chemistry. And I am very sure that our learned speaker, resource person, those they have come from different places, from Japan, from this Saudi Arab, from Dhanbad, from cultivation of science. They will discuss and give a brief outline about this. I especially express my heartiest regard to Professor Otuski for coming over here in this college. I think you have first time coming here. Yes. And also the Professor Bunik, he is coming the second time. And the Professor Patro, he actually happens to be our ex-student of our college. He's coming, it is not first time, he comes previously many times. And Professor Biswadip Banerjee, he also not first time, I think you have come also previously. So I extend my heartiest regard and welcome all these resource persons and all that delegates and participants, especially the faculty members, scientists, and the students from other institute for joining here in this beautiful convention of this emerging trend of chemistry. I think all your participation and engagement will be enriching and helpful for your future journey. Thanks for everybody for joining here. Thank you.
Thank you, Principal Maharaj, for your guidance. We will now felicitate our distinguished speakers, and for that I request our Principal Maharaj to felicitate the dignitaries. I will first request Professor Dr. Joe Otsuki to please come to the stage, who is a professor, College of Science and Technology, Nihon University, Tokyo, Japan. I will now request Professor Bimal Krishna Bonik to please come to the stage. Thank you, sir. I will now request Dr. Bishwadeep Banerjee. Okay. So I will now request Dr. Niladri Patro to come to the stage. Niladri is Associate Professor, Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. So with this, our inaugural session concludes, and this inaugural session will be followed by our 2 technical sessions before lunch. So in the first technical session, our speaker is Dr. Niladri Patro, Associate Professor, Department of Chemistry and Chemical Biology, IIT, Indian School of Mines, Dhanbad, and his title of talk is pH responsive dynamics govern optical insights into drug efflux modulations. And before formally requesting Dr. Patro to deliver his lecture, I request Dr. Kaustav Mondal to please formally introduce our distinguished speaker.
Revered Principal Maharaj, distinguished guests, esteemed faculty members, research scholars, and your students. It is my great pleasure and privilege to introduce our distinguished speaker, Professor Niladri Patro. He has a brilliant academic career. He completed his PhD from Illinois University, Chicago in 2013, and prior to this, he completed his master's degree in chemistry from IIT Delhi in 2008, and he completed his BSc in chemistry from this college, Ramakrishna Mission Vivekananda Centenary College in 2006. In his academic and research journey, he achieved experience at many institutions throughout the world. He did his postdoc. He worked as the postdoctoral associate at Brookhaven National Laboratory, New York, as well as the Massachusetts Institute of Technology, Cambridge. He has also served as the pre-doc fellow at the Institute of Theoretical Atomic, Molecular and Optical Physics at Harvard University. He has made significant contribution to the scientific research. He has published more than 60 scientific journals, including JACS, ACS Nano, Nanolater, JPC-B, JPC-C, ChemCom. Professor Patro's excellence in the research has been recognized through several prestigious awards and fellowships, including Amra Fellowship by Jindal Steel, the Hema Prabha Devi Memorial Prize, the Parin Graduate Fellowship at the University of Illinois at Chicago, the Harvard Smithsonian Astrophysical Observatory Fellowship, and the in VIDIA grant in 2018. His research interests are remarkably diverse and interdisciplinary, covering peroxide for the photo-induced applications, novel metal organic frameworks for toxic gas captures, inhibitor design for efflux pumps and kinases, polypeptides as the therapeutics, and in enzyme catalysis, as well as the development of free energy methods using classical molecular dynamics, simulation, and machine learning. With this short introduction, now I request Professor Niladri Patro to deliver his lecture. Thank you.
Thank you for the kind introductions. Now I think it's okay, right? Okay, so what I'm talking about mostly, I mean, forget about this kind of complicated titles. So I'll be talking about mostly how you have the drug resistance in your body and how one can understand it and what could be done in terms of that. Okay. So before we begin about the drug resistance problems, so our lab was doing for last 6-7 years different kind of research. For example, one of the methods that we developed how we can use polypeptides for therapeutics, and then we have this. Today I'll be talking about this efflux pump problem, and we also have some related to Parkinson disease and etc. All those desires, and then you also have something called enzyme catalysis, which is in between classical and quantum. But mostly these are all computational studies, and we have some collaboration in between, but mostly today I'll be talking about collaborations, not collaboration studies, about computational studies. So for the simplicity, I skip most of the technical part, how we did that, because for master student it would be a little hard how we do that simulations, but technically it's not really hard. Think about something like a Newton's second equations, how you generate the trajectories, and using some classical method and some quantum methods, we did some analysis and study the protein structure, etc. Okay. So the idea about the project was developed from this, like the drug resistance. Most of you know, right, what is the drug resistance. For example, if you go to a doctors now, if you have amoxicillin, so like for example 20 years back you used to have pure amoxicillin, and now if you go to the doctors or any drug store, you will have something called amoxicillin 500 plus 125 clovonic acid. Okay, so that clovonic acid is actually inhibited, it's not a pure drug. So the drug is the amoxicillin, which is the beta-lactamase. Okay, so what is the role of those inhibitors? I'll try to discuss now. And then if we understand what is happening and what is the remedy that one can do. For example, globally now everybody is now concerned that most of the drug is almost end of their life. So that's why you see the combination of drug is now coming into the market. And when we say combination of drug, mostly for a normal disease, not the cancer. Cancer has a different recipes, but normally the drugs comes with the inhibitors. Okay, and if you see that, so drug resistance, typically if you define how is drug resistance in a normal language, you can say when the drugs loses its efficacy. Okay, there are different mechanisms by which the drug basically loses its efficacies that I'll be discussing a little bit, and then we'll go to one of the methods and we try to find out some solutions. Okay, so these are the different ways that the drug can lose its efficacy. For example, initially the drug was designed for a particular target, and over the time the target is actually modulated. Okay, there is something called horizontal gene transfer. Oh, that is very nice. Thank you. I can go here. Okay, so if you see this one is one of the reasons by which the drug is loses efficacy. For example, when the binding structure, so the drug is no longer binds effectively there. There are different reasons why the protein loses its structure. Sometimes it is not only protein, sometimes RNA is also responsible. Okay, and the second one, which is related to this inhibitor I'll be talking about a little bit, drug inactivation by the different enzyme. For example, most of the old classes drugs are like for example penicillin. They are beta-lactam based antibiotics. Okay, and all those bacteria in our bodies that we are trying to kill, they also produce some another kind of enzyme, or basically another enzyme. That enzyme actually metabolize the drug. So that's why what happens is, when you have this amoxicillin in our body, there is a beta-lactamase protein or enzyme. It also metabolize the very amoxicillin drug. So the concentration of the drug inside the bacteria is actually now reduced. So what happened is, now